The idea of Track 2 at its heart is really to build interjurisdictional collaborative teams. It's to build on the investments that we've made at the jurisdictional level and allow you to come together to produce emergent teams that become nationally and internationally effective and competitive. And that's really one of our major goals within EPSCoR. We understand that it takes great teams to do great science. Additionally, it's about building sustainable STEM capacity. And that's the idea of looking at those three golden pillars, not looking at infrastructure, looking at human knowledge or the frontiers of human knowledge, but also looking at the frontiers of human capacity and how we can build that. And we hope that these projects exemplify diversity in all manners, right? Whether it's institution, whether it's HBCUs or incorporating MSIs and these kinds of stories. And the last piece is really helping focus to build the next generation of early career faculty, to create new leaders in these jurisdictions and provide pathways to that. A couple of examples of this is when I sort of look back across our awards. At the moment, we have 48 active Track 2 awards in the program, covering 2015 or the FY 2015 through to FY 20. And out of that, that represents a total investment on NSF's part of $225 million. Moreover, if we look at the incorporation or the involvement, if we look at all 55 55 awards that have been made since 2015, out of that, 26 of our 28 jurisdictions have participated. Of that, 110 universities, colleges, and organizations. And 14% of that, that's 16, are MSI designated. That's huge, and that's a great thing to think about. The thing that becomes important, though, is as you think through our merit review process, we really have you think about, or have our reviewers think about, how each one of these projects are going to be able to create interjurisdictional collaborative teams, how they're going to build as a team, how they're going to build workforce development, how they're going to develop research capacity, how they're going to impact their jurisdictions, and how they're going to interact or integrate these project elements to be the very best of team science that this country has to offer. And so when Loretta asked me to create a panel to be able to showcase the excellence that is Track 2, it was a really hard decision. I had 55 awards to choose from. And out of that, I settled on 8 in the end, but it really doesn't do justice to the 55 awards that we have. It was such an incredibly difficult decision. I could have easily talked about Peter's work on the neural basis of attention. I could have easily had Shunha talking about linking his Track 2 project to massive international collaborations through IceCube. I could have had Ramanis talking about how he's working towards making paradigm shifting work, looking at single cell genes or single cell genomes out of populations of microbiomes. I could have talked about Jersey's work, looking at alternative uses for biochar, and Steve's work, working on genetically informed analytics for crop production. This was an incredibly difficult decision. So when I settled on the 8 sort of that we're talking, or the 8 individuals that will be presenting this afternoon, it really came down to a diversity of ideas, a diversity of projects that'll showcase the full extent of Track 2 projects, to have people talking and describing projects that have finished to projects that are just beginning. And the story that comes out best is when they tell it. Each one of these projects is a unique story from the beginning. And the sort of things that I asked them to do was I asked them to give a 7 to 10 minute presentation where they showcase the best of their projects, whether it's at the very end or the very beginning. And it was really up to them to tell their story. The thing I asked them to do is not just to showcase the amazing science that they're doing, but really think about that these are research infrastructure improvement projects. And at their heart, they're about building these amazing interjurisdictional teams and networks to do great science that will position these teams to become national or international leaders, to help them develop the workforce in their jurisdictions and train the next generation of faculty members. Because the deal here is it's not about the outputs of the project. It's not about the papers and the manuscripts and the grants, and we could easily talk about the hundreds of papers or millions of dollars of grants these produce, but it's about the outcomes of these projects. It's about how they build science, or they create teams that build science and push the frontiers of science in particular areas. It's about building interjurisdictional collaborative teams that cover massive swaths of geography. It's about building sustainable STEM capacity, and it's about building the next generation of early career faculty. And that's the heart of Track 2, and that's what I'm excited to showcase today. And so without further ado, you've heard more than enough from NSF. Let the ambassadors of NSF step forward and tell you what they're doing. And so with that, what I'm going to do is I'm going to bring us on to our first presenter, Ricardo Papa from the University of Puerto Rico. He's going to take it away with the first presentation. Thank you so much.
Looks like Ricardo may have dropped off.
Of course. He got stage fright, everybody, after that introduction. So what I'm going to do is I'm going to switch to our next person on our list. Jamie, are you there?
Yes, I'm here.
Are you able to go out of the gate first?
I am. So let me just jump right into it.
Perfect. Thanks a lot.
Okay. Give me a second to share my screen and I'll be on board here. Again, I appreciate the opportunity to do this. You know, we found this to be a really transformational program, you know, and for all the reasons that, you know, NSF has obviously highlighted in terms of the capabilities and the purpose to build the program, you know, and to really have a, you know, meaningful impact, not just on the investigators, but also on the institutions. So I'll talk a little bit about our Track 2 program, which we just finished up this last year in 2020. And it was called Collaboration Leading Operational Unmanned Aircraft Development for Meteorology and Atmospheric Physics. And, you know, rather than really kind of focus on the purpose, you know, of our program, because we could talk a lot, you know, about the details of the science, you know, due to the diversity of the teams that you have on Track 2 programs. I'm going to kind of really focus on the high level aspects of this and really how it transformed our different universities, you know, both collectively, you know, as a team, as well as individually to many of our teammates. And our basic science question that we were examining was, you know, how can unmanned aircraft or drones, you know, be utilized for, you know, better science, both in meteorology and atmospheric physics. So we had a wide diversity of team members from our 4 partnering institutions spread across 3 jurisdictions that were looking at this perspective from all kinds of different viewpoints. This include engineers, computer scientists, meteorologists, atmospheric physicists, as well as policy experts. So we had both geographers and policy or political science focused, you know, viewpoints, you know, looking at, well, how can these systems actually be integrated to the national airspace, and what is their impact on, you know, the people not only within the U.S. but across the world. You know, looking at, you know, things from micro weather to disaster response to the impacts of climate change. So it was a very diverse program. And so that was one of the challenges that we really had to deal with, are bringing these different things into play, as well at the same time, you know, trying to challenge the current system as it's set up and introduce that paradigm shift where you have these traditional technologies that are being utilized and really not replace technologies, you know, with these autonomous systems, but really supplement them, you know, using, you know, these new techniques to be able to improve the data that you get or increase the quantity of data that you're dealing with. So eventually, you know, starting to struggle with some of those big data challenges that we had as well. And one of the things that we did to make our team successful was bring our team together on an annual basis. You know, this is a, you know, the amount of teams that you're dealing with, obviously, if you look at the number of PIs that we had, and each of those PIs are going to have 1 to 2 to maybe 4 or 5 graduate students working on the project, as well as a number of undergraduate students. How do you make sure that you have a cohesive vision, that all of these students understand the big picture? And how we did that was on an annual basis, bring the team members together in one location, spend one day in essentially a lecture setting, talking about the science goals that everyone wants to achieve, looking at the different perspectives from the various team members, and then conducting, in our case, field exercises, using different autonomous systems that various teams had developed, and going out, taking data, and then comparing it with one another to try to answer some of those fundamental science questions.
Sorry to interrupt, Jamie. I think we're having a hard time seeing your slides. I don't know if they're keeping pace with you.
Oh, let me try to.
Could you reshare and also turn on your video?
Yeah, I'm going to do that. I'm going to reshare the screen here real quick. My apologies.
Also, Jamie, we'd love to see you. We're just seeing a black box now.
Oh, let me fix that as well.
Okay, great.
Yeah, sorry, the hazards of technology. Okay, let me try this here. And the first one that basically set up the science problem. This is our wonderful team that we had from the 4 universities across the 3 jurisdictions. And this is where I left off, talking about the annual event that we held every year. And the original idea behind this was actually to have this hosted at different universities. So we had some of our original planning meetings at one site. And then, once we were awarded the grant, the plan was to move that around to different jurisdictions. But then we decided as a team it made more sense, both in terms of access to particular sites or just the advantage of the campaign that we were running, that we held the first 2 years within Oklahoma at the OSU site. And then we actually held the 4th one outside of the 3 jurisdictions, actually at a Colorado test site. And I'll talk about that in a minute. And then we capped everything off at the end by hosting a symposium that included not only team members across the track 2, but also inviting those from the outside to come and learn from our experience. And that included some workshop participants as well. Kind of going back to the science question, I think this is really kind of the only science slide that I'll really talk about. And I think this culminated and showed the capability of what the team was able to do to demonstrate utilization of these systems into answering some very fundamental atmospheric science questions. So we essentially expanded our team, and using the track 2 funding to be able to leverage participation in a much larger national event. And at the time, this was the largest event using UAS or drones for atmospheric science of its kind, and as far as we know, still remains the largest event, where we spent a week, both all of the track 2 team members as well as other universities and federal partners participating in this week-long campaign to answer basic science questions. And the data analysis from this, even though this was in 2018, so almost 4 years ago now, is still ongoing in terms of publishing the data from this particular campaign and doing additional data analysis on it. So, even though your track 2 may only be a 4 or 5 year program, you'll end up obviously with scholarship that can be accomplished for years to come. Now, what we did do in terms of organizing our team about these different events was essentially build upon a concept known as Hardin's integration ladder, where we started very early with our 2016 campaign, which is our first annual event that we started bringing people together. which was really just getting everyone in the room, sharing their concepts, their ideas that they want to answer, and essentially building upon that ladder rather than, jumping right in and saying, we are a multidisciplinary team because we have multidisciplinary viewpoints, doesn't mean that the science was yet multidisciplinary. So we essentially had to go through a crawl, walk, run phase to be able to make sure that we're all on the same page and we're trying to answer a common science question among the team members. So it's definitely a growth phase. It's something that you actually have to think about and plan for when you're starting to bring the different team members together. Obviously, a lot of this was outlined in our proposal, but we don't even start to begin to think about some of the really tough questions, until you get everyone in the room and you start talking about the different viewpoints and the different problems that you have. Now, this was also facilitated by looking at small group research ideas in terms of how do we bring these small groups together from a social integration standpoint. Making sure particularly that when you have these different viewpoints that you're going to be able to trust your various team members that you're working with. In our case, it was a very organic team, where we had mutual connections that we're able to build upon. But obviously, we're bringing new team members in. So how do you make sure that you're building up this collaborative and collective organism to be able to go through this knowledge sharing process? So that way, at the end of the day, you all have a common set of goals, as well as agreement on what success looks like. And one of the things that we did was track how our team evolved over time, primarily through the connectivity. And so we had a number of different metrics. So these are two examples, one looking at the proposal collaboration, who on the team is collaborating with who else on proposals. So as we're going through as a team, how does that grow over time as we're starting to make those new connections? And you can see the evolution of that from the project start to where we were at the 4-year mark, where we're starting to build those new connectivities and new opportunities for all the researchers on the team, as well as in research in terms of data sharing and co-authorship on scholarship or scholarly articles and papers. And again, you can see the tremendous growth through that, where we start off with a couple of nodes. And in this case, it was primarily myself and one other researcher that served as essentially as hubs. But as we're starting to grow the network and gain this trust with other researchers across the project, then we can start to increase our collaboration opportunities. And that's still ongoing to this day, where many of these team members have built upon these collaborative opportunities for new proposals and new developments. Now, we also shared what we learned with the community at large by being involved in international workshops. Those were workshops that both we hosted and that we partnered on with outside the EPSCoR jurisdiction. And this obviously fulfills, many of the EPSCoR and Track 2 requirements in terms of, making sure that you're integrating yourself into the larger scholarly community. And these obviously, last, long beyond the actual Track 2 project itself. So where we're at now with this, now that our project is ended, what do we do? Well, we're still building upon this, looking at, for example, CERC proposals. We're continuing to work on those efforts with proposals that we submitted, last year and proposals that we're still in the process of submitting, NASA University Leadership Initiative proposals, and expanding our team beyond the original team members within those EPSCoR jurisdictions that we had. So I'll leave it at that. Hopefully that's what you're looking for, JD, and I'll let you take it from there.
Okay, thank you, Jamie. Our second speaker, what we're going to do is we're just going to go through the speakers sort of in rapid fire, so it allows some questions from the audience at the end. So be prepared, my panel. So our next speaker is Ricardo. If you want to jump in, and take it away.
Yes. Okay, I'll share my screen. So I apologize for before. I don't know what happened, but my internet completely bailed on me. Okay. Is it working fine?
I can see it.
All
Yeah,
right.
fine.
Okay. Perfect. Yes. Sorry again. I don't know what happened with my Wi-Fi. Completely everything disappeared. So my name is Riccardo Papa, and I'm gonna talk today about our story. I think that's how it was framed, and I'll put a little bit of everything that goes through what has happened in Puerto Rico since the beginning of this grant, our partnership with MSU and Auburn University, our team, our story, and our science. So we are part of the Genome to Phenome. The name of the grant is Genomic Logic Underlying Adaptive Morphological Divergence. And as you can tell, it's a lot about all these butterfly wing color pattern and morphological diversity across Lepidoptera. So I'm Italian, but I've been in Puerto Rico for the past 11 years, and there is a lot that we have gone through. I'll give you a little glimpse in a little bit. The pandemic is just the last of many things. My lab is based in the University of Puerto Rico in Rio Piedras, and it's made by a quite diverse group of students that are, from Puerto Rico, but as well from the States, Europe, Latin America. And so mostly, I'm an evolutionary biology, and I'm generally interested in the cellular molecular developmental mechanisms that underlie biodiversity in general. So the diversity of morphologies. And our project Genome to Phenome, what it's trying to do is trying to connect, all these characteristics that underlie the wing color pattern diversity in natural population of butterflies, okay? So this is our study system. Of course, this is a short talk. I can go too much in depth of our question, but if I can summarize it generally, what we're trying to understand is really what are the characteristics that makes every individual unique, despite being a butterfly of any other organism. And so this is our team. Actually, this proposal started between the University of Puerto Rico and Mississippi State University. And then it kind of added Auburn University because some member of the original team ended up in this other university. So it started as a 2 partnership and became as a 3. So by default already increase, okay, the capacity. And of course, I want to mention from the very beginning, everything that we have been able to achieve, and I'll give you a glimpse, I think would have not been possible outside, the EPSCoR possibility and what the EPSCoR grant enabled us. I'm very excited in what we have done from the scientific point of view, but also on the personal level. I think, despite being scientists, everybody has his own journey, everybody has unique individual that goes through many different stages and experience in life. And so this is our story, okay? The grant was pretty much awarded in August 2017. We were all excited. I would say we were dancing the Despacito in Puerto Rico to celebrate, but a few months after, 2 major hurricanes just went across the island. I was here, I never left. I was, my family left, they went to the States. I remained here in Puerto Rico. I tried to do what I could, but it was, it was a difficult path and journey to recovery. But there is also a lot I've learned. Puerto Rico, it's not novel news that protest and financial instability has been, part of the life in this island for a very long time. you know, on January 6, 2020, major earthquake started to shake, you know, the south of the island. And actually that night I was staying with my family in a place called Isabella. It was, you know, a little bit after Christmas. And I don't know, I woke up in the middle of the night, 30 seconds before the light went off and all the buildings start shaking. And so now, you know, in March 2020, of course, the SARS-CoV-2 pandemic went worldwide. So this is kind of the journey that we went through here in Puerto Rico, and the partnership with our, you know, collaborator. The way that we built our team allowed us to keep on going. Some of the students left, they went to other university. We started new collaboration. So we were able to continue to work on our project. And so, but this, for example, was my lab the day after, a few days after Maria, all the building was destroyed. It took us pretty much 3 or 4 years to reorganize the infrastructure. All our butterfly were lost. The rearing facility was destroyed. And so, you know, I take a little bit of freedom to say, you know, almost from a philosophical point of view, that I guess, you know, life is kind of a Bohemian Rhapsody. If we can frame it with, you know, a song, there is a little bit of everything. So there is pop, there's rock, there's classical music. I took my, and I'll explain it in a little bit. I'll did mostly the job on me, not to get, you know, down and to find the motivation to start over and over again. And so to me, this island, the beautiful landscape and nature and the sunset, you know, provided me the hope for the next day. And this is something that was very important for me. You know, I'm Italian. I especially during the pandemic, I haven't been able to go back, you know, to see my parents for the past 2 years. And I have a lot of international students here. So I understand, you know, the difficult that the students have to face, you know, during this difficult time. And the most difficult aspect that I actually had to take care is really to keep all the students and lab members motivated and engaged in the science, stay positive. And so what I figured out that was the best mechanism for me to be goal-oriented and make sure that they were seeing the sunset and sunrise every day was to create, you know, manageable group of 2 to 3 students that created kind of a subcluster on a specific project. So project that could give them rewarding outcome in short term, have constant meeting, weekly meeting with everybody, and keep my schedule very flexible. And of course, during this EPSCoR, in all the difficult that we have gone through, you know, we have always kept our collaboration and meetings on a monthly basis together in order to see the improvement. And I think in spite, you know, all these difficulties, I'm very proud of what we have done, not only in terms of number, but in the quality of the science that, to me, the best has yet to come. And I'm very, very, very happy about some of the data that I will present you shortly, which are the latest one we have generated. But the most important aspect is the student that we have been able to follow, to mentor, students that have graduated at the level of, you know, graduate student, undergraduate. We have mentored 5 postdocs. We have been able as a group to submit a large quantity of grants, 17 in total, 9 of which was awarded. And before, you know, the last year with the coronavirus problem, we have been able to organize a lot of like a reach activity and collect an incredible amount of data that I kind of did my homework a few days ago in trying to understand, you know, the quantity of data that we have collected in terms of genomic data, genome, RNA-seq, epigenomic, and so forth. It's about 30 or more terabytes. Okay, Among the fact that we came together as a group is also that, and we learn a lot from the EPSCoR because we have developed a lot of pipeline to analyze our data that somehow became also important for us to contribute to the current problem of the pandemic, especially in Puerto Rico. And we submitted few proposals. We got 2 of them awarded. One is actually recommended for funding from NIH. We kind of put our knowledge into, you know, some sort of like needs for the island in order to follow, you know, all the genomic changes in the virus and basically do some genomic epidemiology in order to understand how the mutation variants and so forth are progressing here in Puerto Rico. But going back to the butterflies, our system allowed to move across different aspects, the evolutionary life history, understanding divergence or morphology as well as convergence, and then go and try to understand the mechanism that promote overdevelopment, the evolution of this coloration. And so the 2 characteristics, again, that allow us to really go deep in basic and fundamental questions of biology are these aspects of a single species divergence, so within diversity of phenotype and convergence between different species. And so the genome to phenome and our project try to characterize the genetic changes and the mechanism. And what we have done, and here I'll present in the next few slides just some of the latest data that we're working, we have been focusing lately a lot on the functional aspect of the DNA. So the rearrange of the DNA and the chromatin over development in order to understand how some of these genes really characterize the development of this color. And here's some of the data that the 2 postdoc, Carolina Concha, Steven Van Belleghem, and 2 graduate students, Edgardo Rivera and Angelo Ruggeri, have lately generated. So, of course, that right now CRISPR-Cas9 have allowed also scientists that work in non-model organisms to test the function of certain gene on non-regulatory DNA or regulatory DNA. In this case, you can see the effect of a knockout of one of these major genes, WNT. On the left is a wild type, on the left is a butterfly without the gene. And so we have tested this gene across different comimetic butterflies, so different species that converge towards the same coloration. And as you can see, the knockout effect is actually have a quite different, provide a different phenotype in each of these different converging butterflies, as well as one of the major species that characterize the Heliconius genus, which is Heliconius errata and Heliconius melpomene. So these genes don't have pretty much the same function. So convergence is not achieved by using the same gene in the same way. And this kind of like summary slide show you that in order to achieve the same morphology, probably these genes have been reused in different ways. So the regulatory architecture might possibly be quite different. And so what we think is that we are only being able to see now the portion of the iceberg that emerge above the water, but there is a lot underneath the water, which is the most interesting part. And so what we are working is really to characterize the epigenomic architecture, so the DNA that possibly have a lot of function in this color pattern development, and we're testing different population of the same species and different species that converge. What we are understanding, and I'll go quite fast across this slide, is that the comimetic butterfly have very little in common. That contrasts everything that has been said from the genetic point of view. The gene, which is the toolkit that create this color pattern, might be quite similar, but the way that these genes are being used is completely different. And this is something that I think hasn't been said yet in evolutionary biology. And so we're doing a lot of analysis on the enrichment of this transcription factor on this portion of the DNA that is open, characterize the intersect of this portion of DNA that is open and differentially accessible with RNA-seq. It's a complex data set that requires a lot of people to work all together. We dissect wings in different sections to compartmentalize color pattern across development and do the same. So what we are finding is that we can map all these different data sets and kind of create a very comprehensive pipeline in order to go after differences that might be behind this switching color pattern. And after we identify these switches, we can start looking in this little region of the DNA and look at what is happening. And what we lately have found is a very small non-regulatory DNA, so non-coding DNA, that potentially is in charge in switching red to yellow, and is very important also for the speciation of this butterfly. We have functionally proved that this regulatory element can switch red to yellow, and we are now connecting the dots to understand which could be the upstream transcription factor that bind to this DNA that then activate some of the major genes that are called optics, that then eventually create the red color pattern in these butterflies. So a long story short, we don't have that much time, but I think, you know, that our science is going towards something that to me is very intriguing, and I always thought that exists in nature, kind of universal link between everything. And so I would say that probably what we are finding is that this morphology are, you know, created by an organized chaos that we can call entropy, and that as biologists we are trying, you know, to decode. So with this, I want to say also that the EPSCoR allow us to also receive more grants for different other agency, and we are building all this data all together towards, I think, some new understanding of biodiversity. And with this, I will stop.
Great, thank you so much, Ricardo. I'm going to move on to our next speaker, which is Julia. Seth, you're actually on deck after Julia.
All right, well, thank you. I'm just going to share my slides. How does that look to everyone?
Perfect.
Great. Well, I want to thank JD for inviting me to share a bit of our research today. We have called our collaborative network the ICE Network, Insect Cryobiology and Ecophysiology, and we were funded under Genomes to Phenomes, and we are right now still in our award period. There are 3 jurisdictions involved: North Dakota, New Mexico, and Wyoming, and we all bring different expertises in genomics, metabolomics, oxidative stress, and bees. All right, so the overarching question of our research is integrating the genetics and physiology of overwintering to understand how these bees are going to be affected by climate change. So is winter going to be shorter? Is it going to be colder or warmer? Is it going to be unpredictable? And based on all of these different forecasts, what can we say about the ability of these bees to adapt or plastically respond to climate changes? We have 3 focal bee species, and each of these is used in agriculture, and 2 of them are native. They have different overwintering habits. So that is the overall science. What I would like to tell you about in this brief presentation are 3 fun things, science things that we have been working on. And then I also have 2 things to talk about that have worked well in integrating and building our collaborative groups, and then a bit about workforce development and infrastructure. So the first science goal I wanted to share with you is a model that our New Mexico colleagues have developed that allows us to predict sites of adaptation and fragmented genomes. So we have genomes for all of our 3 bee species, but of course they are not as well mapped or characterized as, say, the Drosophila genome. And so there's definitely going to be gaps that are involved, or places where we don't know much. And so how to deal with those? And in this simulation, you can see that this Bayesian model takes into account linkage, or the relationship between points in which we have genetic data for those in which we don't. and is able to take a gap such as this one and estimate FST or the divergence at that region, and also the confidence intervals involved. And so we're really excited to apply this forthcoming to the bee genomes that we have. Another fun thing that we have developed is this Maldisi piece of equipment. It's a matrix-assisted laser desorption electrospray ionization. But really what it does is it looks at metabolite profiles. And this is cool because it can do it directly from tissue. So instead of having to homogenize the bees, we can do a tissue section and look at where and what kind of metabolites are present. So here's an example of the output with the bee's head, thorax, and abdomen. And here we're looking at lipids. And you can see that lipids are found in the abdomen, and then where they're found, and also the density of lipids. Another cool thing about this device is that it is made out of 3D printed components and was built by people in our Wyoming jurisdiction. So a third sort of like science story I want to talk about is this model that we're going to make that integrates genetics as well as energy and lipids to produce what's going to happen with bees under different scenarios. So we've been collecting temperature data, air and ground temperature, and of course things related to energy reserves, metabolism, metabolites, genomic information. And we want to use this to be able to predict what's going to happen after winter. Will a bee under certain climate conditions be able to emerge successfully? What will be the fitness of that bee? And so one of the things we've been looking at is bumblebee queens. And bumblebee queens happen to overwinter in the ground. They dig holes. And this model that I'm showing you here looks at what would be the right depth for a bumblebee to overwinter depending on geography and the kind of winters that happen. So it's kind of like a Goldilocks scenario where the bee doesn't want it to be too hot or too warm where it overwinters, because then it will burn through all of its lipid reserves and it won't have enough fat when it emerges to be able to found a colony. But it also doesn't want it to be too cold. So obviously if it's too cold, it might freeze and would die. And so here we have ground temperatures for Laramie, Wyoming at different depths of soil that a bumblebee could overwinter, with this dotted line indicating the supercooling point, or the point at which the bee will freeze. And so we took this information and integrated in a model along with soil depth and temperature data across many years across the United States to look at different scenarios for depth of overwintering and outcomes. So you can see that under a deep depth, there are some regions of the country where a bumblebee would run through its lipid reserves and die before emerging. And then under a shallow overwintering, these are areas in which the bee is likely to die because it will freeze. So we're taking this and building upon it to look at other bees and also integrating genomics. Now I want to shift gears and talk a little bit about what has worked well for building collaboration. So of course we've had over a year of pandemic as part of our grant, and I'm sure everyone has the same challenges associated with that. But one thing that helped us out is that starting from year one, we had a parallel field experiment. So we had each of the 3 jurisdictions was going to set up the same experiment, and we were curious about what would be the effect of latitude on bee overwintering. And this was great because we had to coordinate between the jurisdictions. We had to find and design a project that worked at all sites. We had to get everybody involved from the beginning. And that ended up being a really great thing. And even though we've had lots of COVID setbacks, this really has grown. For example, last winter, we decided to do a winter version of the project because everybody already had a lot of the capacity and the know-how to implement it. And so that was an extra thing that we were able to do at all the jurisdictions. Another component that's brought us together was the modeling component.
Yeah, I just want to check that your slides haven't frozen. They may have frozen on our end.
Oh, really? Well, here, let me, I will stop share and I will reshare. So can you see that?
Not just yet. It's just coming up as started screen sharing. Just give me a second. We still do not see. I still do not see it.
Oh,
Yeah, it's just saying you've started your screen share. We can't see the presentation.
okay. Let me, but you were seeing slides, presumably.
We were right up to the model piece, and then it seemed to freeze.
Oh dear. Okay. Well, let me see.
Just try kicking it up again. There we go. Yep, we're up. We can see maps and a bee and.
Oh,
yep,
wonderful.
yep,
Well, I'm sorry. I don't know what happened.
That's okay.
Is this where I left off?
This is exactly where you were.
Wonderful. Okay. So I already told you about the field sites, but here's the pictures that go with that. So all the people at different sites, here are our boxes that we put up in Laramie and New Mexico and North Dakota. So then I'll talk a little bit more about this modeling aim and how it ended up being a great opportunity to bring people together. So when I think of modeling, I think of it as being very complicated and mathematical, which it is, but it ended up being a really great goal for trainees because there was lots of different variables that we had to estimate in order to put into the model. And these variables involved lots of small scale projects that were easy for undergraduates and starting graduate students to tackle. So just looking at metabolic rates at different temperatures, or here's a student who's testing the ground temperature at a site, and here's another student looking at supercooling points. And as another example of this, we actually ran our RIU summer program, and we had students from that program going through the literature trying to find some of these variables themselves. And this aim used to just be for 2 of our jurisdictions, but now the 3rd jurisdiction has gotten and started joining and participating in this just because it's been really fun and very easy way for everyone to help out in small, very doable projects. So just to talk a little bit about our workforce goals, of course, we are training people, lots of graduate students, lots of postdocs, which has been really fun. We also work with the Nature program, which is exposing high school students from tribal communities to science modules. And we also have a partnership with the Wind River Indian Reservation in Wyoming. And one of our universities, New Mexico State, is a minority serving institution. And we've also done well with our infrastructure. So we recently purchased a state-of-the-art GCMS for a metabolomics facility at University of Wyoming. And then also we have an insect core facility, which is we call the Hive at North Dakota State University, where we've had some incubators purchased from the project. And here you can see one of our early career scientists checking his bees. So if you want to learn more about our science, we have a website and we have a Twitter. And I just want to recognize all of the people that have been involved so far in the project. And despite the pandemic, it really has been a lot of fun. And thank you for letting me tell you about it.
Thanks so much, Julia. So what we'll do, let's move on to Seth. You're up next, and then Nick, you're on deck.
All right. Let's see. How are we doing? Can we see my screen?
Yep, yes. Up and good.
Excellent. Well, incredibly humbling to listen to these stories and to be part of this. So thank you so much for having me today, JD. The story I'm going to tell you today is about the multi-scale multiphysics modeling framework for genome to phenome mapping by intermediate phenotypes. And our jurisdictions are University of Idaho, Clemson University, and the University of Kentucky. And we met through a targeted interest with different disciplines, and of course the modeling and genetics coming from Clemson, a lot of the engineering coming from Idaho with some in Kentucky, and an understanding of cell wall and biomechanics coming from Kentucky as well. And we have a really incredible group of co-PIs and PIs, and there's no I in team. And this is also our external evaluator, who's been incredibly helpful at Purdue. And I couldn't do this talk without saying an incredible thank you to the students. This is a really high student density project because during the summertime and the growing season, there's just such a lot of work to get done. And I'll explain a little bit why that is, but these folks all have these custom-built lodging devices in their hands to cause plants to break, and they have to measure how and why this happens. And so I know in all the jurisdictions we have students who then all get together and fly around to these different spots, which has been a bit difficult with the last year, and they all get together and work on team projects. So it's been really great. So let me tell you a little bit about why we did this project. You know, we've had a pretty good run of the last 70 years in agriculture. You could say the world's greatest environmentalists have been agronomists and farmers, because they have been able to minimize the amount of land being used to produce vastly greater numbers of calories than we ever would have expected. But there's been some problems with that as well. But this is just a laundry list of all the things that we've done in the last 70 years. It's part of a bigger talk that I do on this, but it's pretty compelling that we've been able to basically 7-fold increase in corn yields in a very short period of time. And when we looked at the largest problem still facing production of those calories, and this doesn't just hit corn. You think any crop, whether it's rice, wheat, sorghum, millet, any of the grain crops in the world have huge issues related to losses due to the plants falling over by the performance and failure of their stem. And if we surveyed a range of corn producers from the Midwest, and this was by far their #1 economic loss, I think a 1% improvement of lodging would equate to well over, right? That was with $3 a bushel corn. This is now probably double that, $4 billion annually. So solving this problem has a huge economic importance. This is just in the United States, to not only improving the efficiency of our production, so we don't have to cut down forests in order to grow food, but also to increase the profitability for farmers. Going back, extreme weather events are really bad for lodging, and we're seeing a lot more of those across the globe. And so we are very much looking to grow this project. And our overall goal was to show that we could find this problem tractable. And what we needed to do was combine engineering theory with finite element modeling as the main driver, and that normally look at nuclear fuel rods or wings on an airplane and apply that to a plant stem. And then we used intermediate phenotypes to tie genome to phenome data and map complex traits. And this is quite an unusual series of faculty, and we took quite a lot of time when writing this proposal to even speak the same language. And I'm going to take about 4 or 5 minutes right now to whip through some results, but I really should mention we've had a couple faculty on the project, and I mentioned this, sort of some, when we started, they were junior faculty moving into senior faculty. And these jurisdictions have really grown out now, and we have more and more of these networks thanks to this Track 2 project that really allows us to bring more people into the fold, but also mentor on interdisciplinary work, and that incredible opportunity that allows to tackle big, weird problems like this. This is the problem. These are the numbers. Most stems crease and snap, and you walk through a field when this has happened, and you think of the crop circles that the aliens lay down for us. It's similar to those, but this is actually induced by environmental weather patterns. And this is something that they take on these various failure mechanisms that are characterized by our engineering team mathematically, and then we can try to relate these back to the plant physiology and architecture. And it becomes really complicated. Our program directors probably remember these really complex slides because there was just so many interacting components to this problem that probably are the main reason it wasn't solved for the last 100 years of work in agronomy. And honestly, there was other lower hanging fruit than this problem. But when we start getting to this idea where we really want to not lose up to 20% of our caloric potential a year from grain crops, then we need to tackle this in a multidisciplinary way. And this is very much the driving architecture that enables all these workflows to take place at the same time across agronomy, across the engineering platform, and then pushing this all into this mathematical model that Chris McMahon at Clemson runs in order to try to drag out these genome to phenome relationships. Much of it relies on these custom-built, pushing a plant over so it breaks devices. They're pretty simple, but they're actually really complex when it comes to training students to induce a failure mechanism in the field. And so we have these students running around. They custom build these at a shop in Idaho, and we use them here in Kentucky and Clemson and all over the place to try to capture these data. And some of the successes are really validating this Darling device to predict lodging, and done this quite broadly over the United States now, and been able to have some reliability in the phenotyping method, which is very important. And then use this to identify new genes that are regulating rind strength resistance. And I can't tell you how satisfying and incredibly uplifting this was for the team to identify that this was a genetically tractable problem, that we were able to use genome to phenome mapping to identify key genes that are underscoring the phenotype and explaining it. What this is probably another 20 years of work, but there's a lot of people that can benefit from the successful completion of this project. And this is a lot of the GWAS data, so we can really start narrowing down and picking out these genes, now validating them, building them into networks, and start looking for QTLs and other traits where we could have an incremental impact on that lodging phenotype. These are just some successes. I could tell you a lot about a lot of the failures that we have. We've managed to identify some metabolites that we understand are very important, that are laying in behind some of these genes. And this is a tissue-specific sort of a metabolite that we find interesting for underscoring the phenotype in certain tissues and certain failures. But then we did things such as turgor pressure, which you would imagine would be very important for stem snapping. And this is an interesting example where engineering theory said, no, it's not going to matter. And we were thought so much that it would matter, that the students stayed up for 72 hours snapping stems on a diurnal rhythm every hour, and did this in replicated design for a couple of weeks. And so they were pretty tired by the end of it. And all it did was prove that the engineers were right, and that the cell wall and the thickness of the rind were actually sufficiently designed by Mother Nature that the diurnal rhythm and the uptake of water from the soil was insufficient to influence the plant lodging in but more than a couple %. It was really a very minor influence there, and it was very much daytime dependent. So there's all these layers and layers of information that we're trying to build. It's going to take a long time. And I think the other thing is NSF believing in the multidisciplinary approach and the student-driven approach, and the lack of, I don't know, intuition. It's not just one gene will solve this problem. There's so many different components. Is really gratifying, and it's been really incredible to have that degree of trust, and hopefully we can carry it on to some great things in the next 36 months here to finish out the project. Oh no, sorry, 14 months. Sorry. And I promise the update's coming. We've just been in the field planting like crazy lately, and the rain's been really hitting us hard. The thing's written, I've just got to get it in. So,
no problems. Okay, thanks a lot, Seth. For all of you as well, lots of people ask me about my accent. I am from New Zealand, Seth is from Australia, so this was a perfect example to be able to compare and contrast the two accents. From here, what I'd like to do is move on to Nick, and then Vesna, you're on deck.
All right, everybody seeing their correct screen up there? Seeing a screen?
Yes, we see your screen. Just go ahead and put it in presentation mode and you'll be good to go.
Awesome. There we go. All right, thank you so much for the opportunity to speak here, JD. I want to tell you a bit today about my Track 2 award here. Actually, our Track 2 award here. So our Track 2 award establishes this consortium for plant invasion genomics. The major idea behind this and what we're trying to do here is we're really trying to understand more about invasive species using genomics, bioinformatics, and big data. And then we're also trying to build a large network that spans all of these EPSCoR jurisdictions to really train the next generation of invasion biologists and give them the opportunity to be able to use some of those tools that we're using to answer these new questions. So if any of you guys have walked outside today, which I'm sure almost all of you have, what you've seen is probably at least one introduced species if you looked at any plants anywhere in any of our jurisdictions. These invasive species, they often have maybe no consequence, but sometimes they do have consequences where they can completely destroy ecosystems, they can disrupt agriculture and destroy crops, and they can then cause damage, health effects for humans and for wildlife as well too. Added up in total, this is even an old estimate, this is almost a decade old now, but invasive species are thought to cost just the United States 120 billion annually. And with the pandemic over the last year and a half, you can imagine that the estimate here is just going up right now. So you might think, why invasive species? Invasive species have gotten research attention for the last 60 years at this point. And there's a lot of people that have really thought through the traits that underlie invasiveness and thinking if we can predict what species are actually become invasive. Unfortunately, the major goal that's been neglected in that is really trying to understand the evolutionary forces that underlie this invasion process. And that's what we're really interested in with our grant. Thinking about how different colonization dynamics, the amount of introduction events and the timing of introduction can cause invasions. Thinking about how gene flow between those introduced populations may actually create novel genetic variation that could lead to adaptation. And then finally, thinking about how adaptation in that introduced species could actually lead or facilitate new invasions occurring. Why hasn't this been done frequently? Well, one of the problems with this is it's really difficult to track these invasions. We only get one viewpoint typically of what an invasive species is, and that's the one that we're seeing out the door right now. What would be really nice to be able to understand the invasion process a little bit better is if we could have multiple different viewpoints, starting when the species was actually introduced and moving through time to the present to where it's become a known nuisance or invasive species. One way that we may be able to do that is in plants specifically, or in animals where we could use other collections, is using herbarium records to go back in time and examine populations where these species were introduced, notably closer to the introduction time than what we see at the present day. And we can also use genetics and genomics to track these different invasion events to understand more about their colonization history, where they're coming from, and if there's multiple different invasions, to know if these different introduction events have been able to admix together to produce novel variants, and then to look across the genome and determine what genes actually may underlie invasiveness, and simply determining whether adaptation can actually make a difference to facilitate invasions. So our network is really focused around 5 different hub institutions, and each one of those hub institutions looks at a different focal invasive species. We have white clover, which is the focus of my hub at University of Louisiana. We have Johnson grass, Japanese stilt grass, Russian thistle, and giant salvinia. I'll talk about the different co-PIs associated with each one of these in a couple minutes here. But one of the beautiful parts about invasive species for facilitating this research is people tend to collect them a lot. They're really easy to find. You can go out to your parking lot and then you can make a herbarium specimen from a weed in your parking lot. I had to prune this list for this map up here to make sure it was small enough to actually present, down to 14,000 specimens from nearly 50,000 that we can find online for white clover just in North America right here. But so I wanted to give today a little overview of some of our conclusions from the first 2 years of this project, because we're just ending our 2nd year. And then I wanted to talk a little bit about network building and give possibly some advice that I've accumulated over both successes and failures over the last 2 years building this consortium. So a couple of results to start off with. This is the first big result is thinking about invasions, and not all invasions are composed of just a single introduction event. We have our 5 different focal invasive species, and not one of them was stemmed from a single introduction event. This may be not novel to anybody that's thinking about white clover or Johnson grass that were introduced as crops, but for a species like giant salvinia, this is an invasive fern that forms mats throughout the southeast. It's thought to reproduce almost entirely asexually. And there are populations of giant salvinia that are very benign. They don't seem to increase rapidly in their ponds, but there are others that are highly invasive and seem to spread really rapidly. So when we investigated this question using 38 different chloroplast genomes, this is Stacy Holt's work, and you can see this is his giant salvinia that he's looking at right here in James Beck lab at Wichita State. What he found was there was actually far more genetic variation, even in chloroplast genomes, than what we expected to find. And you can see there's 2 well-supported clades over here, which suggests there's at least 2 different origins of this introduction in the United States. Interestingly, the ones from Arkansas seem to spread a lot less quicker than the ones that are introduced throughout the rest of the Southeast that we have right here from one of these particular invasive lineages. And this is something that we're following up on right now to understand how these different lineages may actually have different invasiveness. We've also noticed that there's genomic properties of these invasive species that may help to facilitate invasions. All 5 of our focal species are polyploids. That is, they've had genome duplications in their genomes that are now 4N or higher. One of these species that we didn't know this for beforehand was Japanese stiltgrass. We didn't actually know if it was a diploid or a polyploid. But Craig Barrett at West Virginia University constructed a chromosome scale reference genome. And it became very clear very quickly that there was this whole genome duplication where over 1/3 of the genes are still nearly perfect duplicates today, suggesting that this has been a duplication in the very recent past. Indeed, when you start looking at transposable elements and using a molecular clock, what you can find is about 1 to 2 million years ago, we see this explosion of transposable elements that corresponds to that genome duplication event, a phenomenon called genomic shock. Now, one of these things from thinking about polyploids and plant species that has directed some of our current thinking is this question: does genomic redundancy facilitate invasiveness? And what are these genes actually doing? Is there some potential for neofunctionalization of these genes that can lead to or facilitate adaptation to novel environments? Finally, I think my favorite conclusion, because it's my research out of this actually, is understanding the role of adaptation in natural selection and invasions. It's thought because it's over such a short time span that natural selection may not play a large role during invasions. So in collaboration with Mark Johnson and my graduate student Simon Innes, we're looking at white clover, and we're recollecting in a bunch of different areas that have been collected over the past 70 years by a biologist named Hugner Daudet, who collected these areas in the 1950s. So we've recollected all these areas from Japan, New Zealand, the native range in Eurasia, South America, and North America. And what we can find out of this is that in an important key defense trait, we see this latitudinal cline that forms in the native range that hasn't changed much between contemporary specimens and historic specimens. But in the introduced range over that 70 years, what's happened is that these clines have evolved to look more like the patterns that we see in the native range for that key defense trait. What this suggests is that adaptation is constantly occurring in these invasive ranges, and it's occurring in a specific way, sometimes towards the patterns you see in the native range, but towards the specific novel environments that you encounter. In Japanese stiltgrass, we have a lot less historical data on this, but what we can see from our barium specimens is that there are clines in key functional traits. For instance, there's a cline forming in the ability to produce awns, which are these little bristles that occur on the end of seeds here and are thought to be involved in burial of these different specimens and proper germination, where there's far more awns in the northern latitudes than there's in the southern latitudes in these individual ranges. Okay. Those are some of our cool conclusions that we've just started off with in these last 2 years that we're following up on here. But what I want to put out here is that each one of these focal species has had something really interesting to contribute, and that when we put all these focal species together, we make really interesting conclusions about what invasions actually are and about the particular invasion process. sometimes where things are unique, but sometimes where we're seeing generalities that are forming in these different focal species. Our other goal was to train a new generation of invasion biologists that are both literate in genomics and bioinformatics. And we started off actually a Twitter conversation a year before we actually formed this group in general. Our project formed off a bunch of people who wanted to do a bunch of genomics and bioinformatics networks throughout the United States to demonstrate common NGS methods, provide some hands-on training, and then focus on some collaborative research. We've taken all this and our PIs together have created this hub and spoke training system where we have 4 different hubs located in 4 different EPSCoR jurisdictions with different regional PIs. And then each one of those hubs then has 4 regional PIs and then 4 local PIs for each one of those regional PIs. And we run boot camps for these regional PIs, and then we run them in collaboration with the regional PIs for local PIs the following year here. In such, we're training a bunch of different PI level scientists how to use genomics and bioinformatics. A lot of them have only been exposed to Sanger sequencing methods before. Currently, we've been training 12 different regional PIs, and we've been recruiting our local PIs. We're up to about 20 local PIs too. And we're currently encompassing 15 different EPSCoR jurisdictions with our network they have here. But we've become more broad from those original Twitter days. We have these training networks where we're training regional PIs. This is my particular hub right here. But we also have a large undergraduate training program during the summers where we're training 24 undergraduates focused in 14 different labs this summer. We're also building these collaborative research projects. With Roxy Keller here, I've been working on garlic mustard with one of her graduate students as an invasion front. With Paul Kim, I've been working on transcriptomics of white clover during drought. And with Sarah Fuentes Soriano here, we've been collaborating with Marcelo Sternberg in Israel to look at the evolution of camphor weed. So this is developing collaborations and building out from these networks themselves. And we found a new role for ourselves too, is that to structure the next generation of this field and provide a framework for this field. So we've written perspective articles on how we think our data can contribute to these new frameworks and to what questions they can actually answer, what research needs to be done. Every year from here on out, starting in this August, we're holding in-person meetings. Cross your fingers for COVID, for no COVID problems associated with this, down in Lafayette, Louisiana. And we've had interest from almost 150 people already for this event, and we have to limit to 75. So we're moving. I have a couple pieces of advice for people trying to build these networks starting out, and for just thinking about how network development actually should work from our successes and failures that we've had in building CPING over the first 2 years here. First of all, the most important thing that I think anybody would agree with here is developing a proper communication system first that is coordinated, that is decentralized and hierarchical, and that's not overbearing for all these people that are training in this network but may not be involved on a day-to-day basis. We've really successfully used Slack frameworks for this with different channels for different groups and for establishing little clusters of collaborations. Second of all, and this stems from our Twitter days, is make your network inclusive of all comers. This may not necessarily be specific to EPSCoR. We have a lot of people outside of EPSCoR that's collaborating with our group too here to develop other collaborative endeavors, given that they're interested in these same questions too. We advertised widely, we go to listservs, and we're still working on ourselves to figure out how we include more diversity in our network in general. And for us, it's really worked well to go institution to institution to places that we weren't reaching otherwise, and trying to actually reach out to professors that are there. And finally, I can say this as a young early career PI here, don't neglect the administration and plan for the time drain that occurs from establishing these different networks, because the organization and oversight of these networks takes an incredible amount of time to get them off the ground. And if it's decentralized communication with multiple of your PIs working with you, you can make this bearable, but you have to really think hard about how you want to structure this from the outset. The last thing I want to say as well is for program officers, for any of the universities, and for any of the established PIs out there, but occasionally step out and let the teenagers drive the Ferrari. We are almost all early career professors, and we're training all almost senior professors for our regional PIs that are out there that have kind of been left behind by this genomics revolution. And I think we've done really good so far. We've gotten 8 grants funded in our first 2 years already. We've done really strong work on each of our focal species in here. We are the geriatric millennials that we referred to in that Times article last week. So I guess let the geriatric teenagers drive the Ferrari. With that, I want to thank everybody in my group here. These are the early career researchers that are really integral to this project right here and that we established on Twitter initially. Craig Barrett, James Beck, Maryvette Laspis, Michael McCain, Aaron Sigal, and then our program manager, Brittany Sutherland, and Danu Ramachandran, who is our postdocs currently on this particular grant.
Great. Thank you so much, Nick. Vesna, you're up next.
Okay, I'm gonna share my screen here. Start to broadcast and get to the right app here. Here we go. Thank you for this opportunity to present our work. And I want to say that thank you, EPSCoR, for allowing teenagers, well, we might not be teenagers by age here early in our project, but we are in a sense of that we are what this grant has allowed us to kind of really venture into this new field of physics. Hopefully that's going to be apparent, or at least to try to merge two fields that never talked before together. And it's been really fun. So I'll be talking about our progress that we have made in this using basically big data techniques, numerical techniques to allow us what we call a quantum leap, meaning to allow us to control quantum systems and quantum materials, real materials. I'll explain later what I mean by these materials. And this has been a really wonderful collaboration between Brown, our team at Brown University from Rhode Island and Dartmouth and University of New Hampshire. And so what are we really trying to do here is to, as I said, the main goal of our collaboration, okay, good, you can see my highlighter here, is to address really two challenges in quantum science, which is, you know, in quantum, everything is complicated. I'll show you a picture why things are so intense or so impossible to control. There's something inherent about the system that tells you that you just cannot, you have a very limited knowledge about quantum system. And so how do we identify what this complicated thing is? How do we control it so we can use it for very useful applications? And then also imagine now that even a simple system is difficult to control. We don't understand how, we don't understand what it is exactly. You can only quantify it by what is not, okay, in kind of classical sense. And then, you know, take a real material, right? And then try to predict its properties and also how to use them. This is really, really difficult, okay? And so why, I think this is much easier if I just show you this picture here instead of why is it so difficult to have this, how do I say this, quantum, why a quantum system so difficult to control and identify? Well, there's a little sketch here, is that if you look inside of this beautiful properties that we're supposed to use and make these amazing machines, is that, you know, each of these little red balls, which is an electron here, it's connected to either one or two or, you know, many. They're what we call in jargon entangled, okay? So, and if you try to measure something, okay, right here in this part of your sample, what you actually read out depends on, you know, this mess here, okay? And this is where it's super difficult to actually deal with this. So ideally, we would really like to know what all these green lines are. How do we break them? How we establish them? And so that, you know, when we measure something here, that we know what it is and it tells us, you know, everything about this, okay? So that's kind of a big picture of what, you know, this whole effort is about. And it requires actually, because of this complexity, requires kind of expertise in two very orthogonal fields of physics, which is, you know, people who study materials, what is called condensed matter physics, which we're we have a strong expertise at Brown, and then also people who have studied what is called quantum control, where you actually take weaker interacting, less complicated systems, so to speak, and you really try to control them and identify them with this very well controllable way. So this goes by the name of what is called quantum information science. And this is where Dartmouth has its strength. And we've been really, through this project, enjoying to learn each other language, our challenges. And so I'll talk about one of our main project here at Brown that, of course, is enabled by our collaborator at Dartmouth as well. And this is, so when we try to get this complexity, what we want to do is, our idea is to look at inside of these little red arrows, electrons, there are nuclear, and they carry something called spin. It's that very property that allows us to have, for example, MRI. So it's something that we know how to, measure. And so these nuclei, these are these purple little spins here, they're, everything that the electrons, those green lines that I showed you here, they're kind of attenuated, okay, if you think about it, because the green lines connect the electrons. These guys, nucleus, they're weakly coupled, they sit inside of the material. And so because the things are so complex, then our idea was, well, why don't we look at the simpler system, which is this nuclear system, which only kind of muffles this and interacts, these two nuclei, they interact through this kind of electronic, this little wiggly line is basically what these electrons are, and we don't know what they are, but we'll try to simulate this interaction. So the idea is that we're going to basically look at how the these nuclear spins are coupled through these electrons, and then we can use all these numerical efforts to actually make these simulations. And this is quite different approach, because what people have done before is just to kind of maybe look, nearest neighbors, weak interactions. We actually, using these numerical methods that we developed, we can actually look at basically couplings that are, long range, short range, change basically these electronic states here. And it turns out what people have done before, it's that these kind of weak interactions, they can only kind of shift the resonance. So we are actually looking at something that we can measure that it's very different than what people have done before. So we basically, instead of looking, we're looking at the whole, this is actually simulated signals, something that it's called the shape of the echo, okay? And so what one does in an experiment, it sends a pulse and then, let these nuclear spins evolve and they interact with everything in this material, and then we detect it again with the same pulse. And so depending on how what your probe is, or this pulse here, and also on the strength of interactions, you get very different signals, okay, outputs. And normally in normal experiment, you actually aim to see something like this, very symmetric. That's what it should happen in a simple system. And people have seen these kind of echoes here when you increase the power, or you increase, system is more complex, and they usually associate that with kind of trivial effects of, you send too large of a pulse, you kind of, this is heating or so on from the pulse. And because it's so complex, nobody ever examined it. And it shows up in many very exotic materials that we study, so-called topological materials nowadays, over and over again. And so we've been able to actually look into details of this. And, this is a physicist making a model. So if you ask me what went exactly into this wiggly line, well, it's here. And then, you just need each of these little line actually becomes a different equation and different integral and so on. And so once you do all of that, then, there's kind of a loop of how we do this calculation. So what we're actually doing is we're literally simulating the evolution of the nuclear spins, or the signals, under the influence of this pulse. And then this alpha is basically changing the how these electrons actually interact, and all these different formulas, and how an isotropy of these interactions, and what we can then, and we can simulate these spectral shapes. And what is actually what we've been able to do by using basically machine learning techniques, okay, to simulate these echoes and simulate this evolution of this huge system is to, we've been able to actually reproduce something that's been seen and also identified almost as a, artifact of an experiment in a lot of NMR experiments in these topological superconductors, topological materials. And it seems that this has a promise to basically, given the fidelity of these simulations, that we're going to be able to actually tell what this, electronic, what is the Hamiltonian, meaning, kind of, what is the, biometric passport of our electronic system. So we'll be able to actually, by looking at these trivial experiments or the effects of these, how do we excite a nuclear system, that we'll be able to kind of reverse engineer Hamiltonian. This is something, or what these collective modes, these, red, those green lines that I showed you earlier between electrons are. And this is something what, every physicist dream of knowing about complicated system, because once we know that, then, we know how to control and use the system. And so this is kind of been a big picture of what we're doing. And again, there are other, another example is to, we've been able to, there are very peculiar, if you can see my lines here, there are very peculiar states that one can use to make supposedly a quantum computer, but nobody knows how to manipulate these states. And again, we've been actually, we just have a preprint where we actually propose a very specific way of manipulating nuclear spins again. And they would actually cause, that manipulation would actually produce the change in conductance. This is, electricity, right? It flows through your device. And again, it's numerically proposing a way of how to actually manipulate spin in these devices so you can actually use them for some, quantum register. The goal is to extend this work again using these machine learning techniques to be able to, again, devise very complicated pulse sequences of these nuclear spins, how to evolve them in time, and then to be able to actually, again, build a probe of these very complicated states that can arise in these, what's called topological materials, insulators. I don't have a lot of time, but I just want to tell you another effort that is, again, from Dartmouth side now. So it was basically using reinforced learning for quantum control in magnetic resonance. So what is quantum control? It basically means that, you can send some stimuli, which we call, again, a Hamiltonian, to your system, that it's weakly interacting. And then what that would allow you is to evolve your system. This is what this word unitary transformation means. You can evolve it in time to give you an effective Hamiltonian that you desire, okay? So what that requires, right, this control here means that I need to apply a bunch of pulse sequences. So some experimental stimuli needs to be sent in a very controllable way. And then again, what our team has been actually using is this called, in collaboration with collaborators from MIT, is to use this reinforced learning for quantum control, where you basically, your state that you start with, your state this algorithm actually is your propagator. Action is these RF pulses that we are applying to manipulate. And then you can actually see what the reward in this algorithm is, which is given by something called fidelity, or how, the evolution that you apply, how well does that describe the effective Hamiltonian that you want to reach. And so you can use these kind of algorithms, again, to optimize what pulse sequences you need to use to give you this, quantum control, meaning put your quantum system in the state that you want. All these numerical methods, again, are used, for example, what's called noise spectroscopy for dynamical decoupling, meaning if you want to make a qubit, this basic unit of a quantum computer, the problem is that perfect cubic here in a diamond, which is a P1 center in diamond that has a spin you can control, it's coupled to environment. And that causes decoherence and limits the time, useful time that you can use this system for quantum, in quantum operations. And so what a group of our PI, Ramanathan and Dartmouth, has been doing, again, using these numerical methods to actually see how to design these perfect pulse sequences or long, just look at these graphs. So how to design these sequences so that you can actually extend these coherence times. And there's an example of where this was an experiment, and that's a simulation. So again, you can simulate what you have to do, then you go and measure that, and you can see the two agree, and that can be used then again to design these very complicated decoupling sequences, more complicated qubits. And I think I'll just mention one thing that we've been working actually between my group and Shekhar Ramanathan, where again, it's this idea of learning each other's language. We had a very complicated quantum state that we've been studying, but it's a very complicated, to use a technical word, tensor order. There's no measurement that you can do that you can actually exactly determine its properties. And so what we've been trying to do is to something what's called spin squeezing, which is a very complicated technique that's used in quantum sensing to actually allow you to really push the limits of any measurement that it's determined by laws of quantum mechanics. And so that particular technique requires some very complicated pulses that we thought if we do, we might be able to actually see this elusive quantum order that we've been studying. And so again, we've been designing, and sure enough, we've been trying to see something. And these little curves here are the simulation, and basically shows you that if your system possesses certain properties that we assigned, you can actually see that this squeezing parameter actually shows the difference. And so there was no measurement in classical kind of measurement that we knew of that's actually capable of resolving or telling you exactly what this physical state is. But if you apply these this kind of quantum measurements techniques, but not in order to actually do this perfect measurement, but actually use just that in order to see the changes in your system, okay? It seems to work, and it gives you really, really, really good probe of, physics of your system. And just to end that, it's very difficult, as everybody pointed out, for people from different disciplines to learn each other's language. And again, we've been really kind of working, what this funding has really allowed us to actually put a significant effort into that. And on educational front, we had a quantum winter school that was dedicated equally to both of these, or even kind of preaching this thing that we need to combine, that there is a lot of potential in combining this quantum information techniques to materials and how to adopt them. And then we've been also having at the machine learning science hackathon, we had this NMR challenge, meaning how the machine learning can be used to really to educate you about NMR or magnetic resonance techniques and control. And also this would be an open source soon, have this complete simulation for the NMR observables. And it's quite unique because there are a lot of programs out there. They either simulate, spectra that you get in chemistry, because that's what they care about, or they simulate how to do this quantum control. And this is actually a first software, Python open source, that would actually allow you to do both and actually put two together. So anyway, sorry if I went too much over time, but thank you so much. Yeah.
Thank you so much. So for everybody, we still have 87 participants online. I am going to have both Amy and Aaron present. I've spoken over with the coordinators and they're okay with staying for the extra time. I do fully encourage you to stay for both Amy and Aaron's talks, because they're also doing excellent science and have some interesting things to share. So with that, if you have got time, they will be done on time. So I presume we'll be finished at 4:20. Amy, if you wanted to go ahead, please take it away.
All right. Not sure if you can see me yet.
Vesna, you'll have to stop.
Sorry.
Got it.
I apologize. I did not see your text, and I'm really, yeah, sorry.
All right. Hopefully you can see the screen now. So thank you to JD and to the organizers for giving me the chance to talk about the first year of our EPSCoR Track 2 project, which is named the Aquatic Intermittency Effects on Microbiomes and Streams, or AIMS. It's a collaboration of 8 different institutions. The University of Kansas is the lead institution, with Haskell Indian Nations University, a tribal college, Kansas State, the University of Oklahoma, Idaho State, University of Alabama, University of Southern Mississippi, and University of Mississippi. We cross 5 jurisdictions and a number of different collaborators who I'll kind of have pictures of them mentioned throughout here. Intermittent streams, to kind of get a good picture in your head, are those that do not flow permanently. So this is the same stream, one of our field sites out at Konza Prairie in Kansas. And you can see here the stream is disconnected into a pool, and Jess, a grad student, is putting in a gauge for us to see how much water's in there. And this is just 2 weeks later, just a very little bit of the same gauge is popping out. So this stream just changed dramatically in the short period of a couple of weeks over the summer, where the pools started to connect and become less isolated and look more like a perennial stream. And so we were funded under the theme of harnessing big data to solve problems of national importance. And the challenge that we identified and articulated in the proposal was that these intermittent streams are really abundant across every region in the United States. They account for somewhere on the order of 50% of worldwide stream miles. However, quantifying and understanding how they affect water quality is an area that's relatively unknown. And that's due in large part to a lack of physical infrastructure, sensors and monitoring equipment that are positioned in these intermittent systems, a lack of methods for studying things at the terrestrial aquatic interface. A lot of times in our field of ecology, people are comfortable in one environment or the other, but rarely in the interplay between the two. So putting some emphasis there, as well as in a lack of training in team science and using big data tools to be able to parse out some of these large sensor data sets and some of the genomic information we'll get from the microbiology. And this is an issue of national importance because it's at the center of a policy debate that's been in the news regarding the changes to the Waters of the U.S. rule. This was a rule to protect surface waters. It was replaced in 2020 with something called the Navigable Protection Rule, Navigable Waters Protection Rule, which rolled back these protections for intermittent and ephemeral streams. And there are several current court cases involving stage tribes and environmental groups that are in the air right now trying to resolve what the ruling should be around this and the EPA's role in it. And so our solution to this problem of national importance was the AIMS proposal, which was to create research, education, and workforce development goals to create a network of highly instrumented field sites to generate big data on these intermittent streams in terms of their hydrology, microbiomes, and biogeochemistry. Those are our 3 kind of big research themes in the group. To train scientists from diverse backgrounds and collaborative and team science approaches, and to provide workforce training and tools integrating these different data streams, including microbiome, sequencing, environmental sensor, and other sources of data. So our EPSCoR Track 2 funding will establish 9 intermittent stream research sites with a lot of different sensors in the outlet of the watershed, as well as integrated throughout the watershed for continuing work on these types of streams during the project and hopefully into the future. We'll train 2 postdocs, 11 graduate students, and over 18 undergrads in these team science approaches, leverage existing programs to increase data training and use of big data, and to train faculty in building inclusive labs and offer training support for diverse students. And so this is our team. Our natural habitat is Zoom, having only really launched in the era of COVID. So we have never actually been in the same, all of us together in the same physical location. But despite that, we've managed to find ways to kind of navigate this new normal and get the project we hope launched onto a good starting point. And so the overarching objective of the project is to really quantify how these intermittent streams affect water quality so that we can inform policy better. better regarding them. And the scientific question that integrates all of this is asking how physical drivers, things like climate, hydrology, and watershed characteristics interact with biology, microbes, and their chemistry of the environment to impact water quality in these intermittent streams. So we have a diverse team in terms of their expertise, and some are more in terms of hydrology, some are more thinking organismal, especially about microbes and some macroinvertebrates, and then some of us that are integrating some of that with biogeochemistry and trying to learn how to all speak each other's language is been a big part of the first year. So our research goal is to build out this collaborative network of well-instrumented research sites where we can test our question. We design ourselves within 3 regions. So we have the Mountain West, which is in Idaho, 3 field sites, the Great Plains and the central part of the US, Kansas and Oklahoma, and then the Southeast, which is Mississippi and Alabama. So those are our 5 jurisdictions. And as you can see in the graph on the bottom, all of these states have a high proportion of their stream length that is classified as intermittent, including Alabama, where there's a fair amount of perennial flow, but 50% of the total length of streams in Alabama is intermittent, yet there are 0 gauges in Alabama, 0 physical infrastructure that would allow us to be able to study these intermittent streams. So we're building that infrastructure to allow for more. And even in a place like Kansas, where we know they're very abundant, very small proportion of the overall infrastructure, 2.5%, gauges are on intermittent systems. So this is a big blind spot in the monitoring systems that we have designed already. And therefore, we don't have a really good understanding of the role of intermittent streams in impacting water quality. And so because we're in year 1 and because we're in the phase of getting sensors out right now, we don't have a lot of results to share, but I do want to talk about what are some of what I think of the distinctive attributes of AIM. So we're a field-based project. Our data don't exist. We're in the process of making it, installing this physical infrastructure and collecting samples. And so we've focused on that a lot in our first year. We've also focused a lot on what we're calling a project-based graduate cohort model, where we've had coordinated recruiting, and we'll have team science training for all of the students that will be integrated into the project. So we had 11 funded positions across 7 institutions that we recruited in a coordinated manner this fall. And next fall, coming in to fall 2021, all of the students that are onboarded onto the project will take a cohort building team science class with me. And I remember writing this into the proposal in January 2020, in the before times, and thinking, I'm not exactly sure how we'll pull off a virtual cross-institutional class. And at this point, I think we all know how we can pull that off with all the technology we've been doing with Zoom and the ability. So that the pandemic has really allowed us to see and build capacity and how to do better. And I'm really excited to teach this class. We'll focus a lot on talking with the students about conceptual models, about taking some of their ideas and how they relate to the larger project and what we're trying to do, and hopefully make them fluent in the conversation around the project, as well as give them some tools to continue to work collaboratively throughout their careers, and some scientific training, as well as some diversity, equity, and inclusion training. We're also building out what we're calling on-ramps to data science. This is our signature workforce development program. And we're partnering with the Carpentries, which is an entity that does software and data science training workshops for novices. And we, as part of our workforce development, are training 36 new instructors, so 9 a year across all of our institutions. Some of our institutions have 0 Carpentries instruction right now. And so we're also teaming, one of our partners is a data librarian on our project, and her research is geared at understanding the best practices for capacity building in establishing a Carpentries culture on a campus. And so she's been really wonderful to have integrated in with the team and helping us figure out where a specific institution is on a continuum of 0 to having a very full-fledged Carpentries kind of instruction culture, and then will help us continue to develop that aspect of the project over time. We'll take our sensor data and develop content based on it and offer workshops at partner institutions that are just starting to try to get Carpentries instruction built at their institutions. We also put a huge emphasis on early career mentoring. Half of our PI team is early career. And so we've really tried to think deliberately and intentionally about how we can support their careers through this project. We've established what we call catalyst data sets. We define this as the data set that a early career person is really excited to build out for a proposal. So it's not supposed to be a territorial thing in any way. It's not saying only you can work on that, but it's more of a, this is where we think, you know, you're really excited to be thinking about your next proposal and what you want to do with what we call the core data set of AIMS to be able to launch those careers using AIMS as a platform and as a structure. So I've had conversations with all of our early career researchers about the types of catalyst data sets and future proposals that they want to do. We've also been creating resources around mentoring for undergraduate and graduate students to share as a group. And we are also designing something we call the Early Career Pre-Batical Program. This is designed to have a couple of weeks for our early career people to be able to go somewhere. And it's really designed in part to be able to take that trip and build it out into an EPSCoR Track 4 proposal. So something that can kind of help launch their collaborative network outside of the AIMS grant as well. So our project, as I mentioned, exists wholly within COVID. So that has largely been the challenges that we've had in this year. And we deliberately designed the first half year or so as a planning phase because we knew it would take time to get our equipment ordered and installed. And I'm glad we did that because it has been a needed kind of window to be able to ramp up, especially given the extra challenges associated with this year. With site selection, there was very limited travel. We couldn't get out in the field, or we had to take multiple vehicles to fields for COVID rules. Equipment purchasing has been very slow. Back orders of many supplies, as I'm sure many of you know. Hiring has brought extra challenges. The silver lining has been that this has really given us a lot of time and ability to converse about the standardization of our protocols and data management. We really feel like we've got that maybe better nailed than we would have had we been all traveling and kind of under academia as normal in the last year. So we're feeling really good about the launch of the data collection phase this year. I will say graduate student for us, recruiting was extra hard this year. There were no in-person scientific meetings, so being able to go and meet students and bring them into the project or get them interested in it was hindered. For most of our institutions, recruiting visits are very important. Bringing people into our institutions and convincing them that this is a place they can envision spending 5 years of their lives. Those were all suspended, so that also really limited student interest. And we had multiple budget issues at many of our institutions, which I'm sure is also the case for others. And in general, the pandemic affected our team. We had a lot of early career people, and many of them had kids at home, and so that really limited their ability to engage as well. Despite that, we had a really good ability to build out research infrastructure this year. We had workforce development. We filled all of our carpentries instructions. We got 12 students recruited to the projects. We got an extra student because we had a successful GRFP applicant, which was a really good first early win. We've hired all the people are in place. We've managed to keep our goals around broadening participation with our number of women on the project at 50%, and partnerships with Tribal College, Haskell are ongoing, as well as with an HBCU. We've got a lot of interjurisdictional collaboration building. By design, our teams are cross-cutting within our region, so we have representation of different research types on all of our various regions, and we're starting to build out this idea of affiliates, so people that were not originally on the proposal but want to be affiliated with the project to use the infrastructure, and how we engage around that kind of interest. We've had a lot of purchasing of stuff and stuff going out in the field, as well as development of proposals, and we're all really excited for a hopefully somewhat normal field season coming up very, very soon. In like the next couple of weeks, we'll be launching a lot of our recently purchased equipment out into the field, and so all of us are really excited to get back out after the last year of really strange times. So I appreciate your attention, and I'm happy to address any questions if we have time later. Thank you.
Thanks so much, Amy. And our last presenter for today is Aaron. So if you want to take it away, off you go.
Thanks, JD, and great to be here. I appreciate everybody hanging in there. We are in year 2 of a project that we're calling NSPIRES. This is a joint venture with University of Vermont and University of New Hampshire, with my co-PIs Anthony D'Amato and Scott Ollinger. Like Amy, we've mostly been working under a pandemic, which has made things a little more challenging. Also, like Amy, we were under the big data grand challenge. And so our framework was bringing together diverse disciplines to work on an issue that has relevance for both science and decision-making. The whole idea here that I'll briefly touch on, and we are organized around 4 central research themes: ecological models, informatics, advanced sensing, quantitative reasoning, and then basically the idea of building out a digital forest, big data framework that supports science as well as decision making. I think a key part of our success early on has been partnering early and often with a wide group of agencies. This has included large NGOs like NatureServe or Project Learning Tree, federal agencies like the U.S. Forest Service, large private forest landowners like Weyerhaeuser, service providers like Esri. Our evaluation is through the implementation group, and then finally we've partnered with the Wabanaki community to round out our tribal science. In theme 1, a big part of this is building out. So we are organized around themes. Theme 1 is centered around engineers, electrical and mechanical, to develop research grade instruments that cost at $100 or $200. And basically we're trying to fill the black hole of this northern forest region where there is no real research infrastructure or even cell phone access. And so working with them to kind of address some of those knowledge gaps. Theme 2 is working with computer scientists and remote sensors to basically develop a data-driven framework for housing the sensor data, as well as doing site selection, as well as filling the gaps in between our research sites. Theme 3 is primarily looking at ecological modeling. So how do we use the current sensor information as well as the maps provided by Theme 2 to ingest into large-scale ecological models to make forecasts of future conditions across a range of scenarios or future conditions. This is really the integrator theme that's taking inputs from themes 1 and 2 and then feeding back information to those themes to refine their processes. And finally, this is the basis largely that we're integrating our educational piece with our high school grade science teachers. Finally, theme 4 is quantitative reasoning and context. This is working with STEM educators as well as STEM researchers to basically develop the idea of bringing together critical thinking as well as quantitative reasoning skills to help high school students think about not only environmental data, but do more hands-on curriculum, working with actual sensors or models actually in the field. I think, as you've seen, these track 2s take a lot of different spins. I would say that our track 2 is probably a little bit larger than the norm. We're about 67 participants, equally spread across the 3 EPSCoR jurisdictions. One thing I'm quite proud of is that half of our faculty is early career, and I feel that this track 2 has been a great way to bring them not only resources to work with, but as we see in a pandemic world, new networking opportunities that allows them to build important relationships. As Amy highlighted, recruiting of students has been a challenge, or even supporting ones that are existing. And so we've had to work hard to make sure that all students, undergraduate, graduates, and even postdocs feel supported. I think a big part of what we're doing, and this I know is part of a track 1, but I think this is really important and what helped us kind of hit the ground running, as well as survive the last 14 months of kind of a virtual pandemic working remotely, is a very detailed living project implementation plan that I just went in and updated yesterday. This includes a variety of your usual strategic materials like logic models, SWOT analyses, stakeholder matrices. We've also thought about communication reports, as well as kind of research milestones and assessments. I think the biggest part is basically creating a one-page document that all of our researchers, whether they be students or faculty, can quickly find things such as our shared calendar, our team roster, our government document, the anonymous feedback form that has been helpful for making sure that everyone is is feeling comfortable and included in this project. I think a big part also of what we did in year one, as we were all kind of working remotely and virtually, is it's hard to work within a jurisdiction, but it's even harder to work across multiple jurisdictions, even when you're in the same geographic range. So we spent a lot of time focusing on team science and team building. This was a facilitated exercise that we did at our last virtual annual retreat. Basically, we divided by Zoom into different teams, and we all came up with different team visions that we shared and discussed. Our hope is that we can revisit this over time so that we see how things converge or not converge, and where the central themes and how people feel included in this overall effort. As you can see here, even though we started off with that conceptual figure that I started the presentation with, all the different teams kind of interpreted that as well as viewed that differently based on their interactions of who they're working with. I think this is important, particularly as we continue to work in a virtual environment. I think a final thing, or another important thing that we have also done is collaboration happens among faculty, but it should also happen across the institutions that are being supported by the track 2. So we formed a tri-jurisdictional institutional advisory board that is met throughout the course of the project. This brings together deans, vice presidents, provost, the people that I think really need to understand how the early career faculty are coping and dealing with the pandemic, as well as trying to find mechanisms that create collaborative opportunities. Through this advisory board, we've identified priorities across the different institutions and are now working on finding things that will help sustain this effort well beyond after the grant ends, even though we're still just midway through. I think a lot of this has resulted into outcomes, and I appreciate that track 2 is outcomes driven. I think obviously year one is tough of getting everyone on the same page and onto the same team and sharing that vision. But if done successfully, I think it can result in a high increase in productivity, particularly when you have a large yet engaged and diverse team like we have on Inspires. As you can see on the left, our publications has increased dramatically, while the research funding has also increased quite dramatically, with several of these being interjurisdictional collaborations. I think this has already been brought up, but it's new to me. Just science doesn't happen through email and typical websites anymore. Many of our students and early career faculty are on Twitter, Instagram, Facebook. This is how science gets communicated with not only the research community, but the public at large. And so we've gone through measures to engage our faculty as well as our students to help populate our social media and make them current and try to engage a broader audience than our current team. So I think overall, despite the pandemic, I feel there's been pros and cons of all working virtually. These projects are hard, I think, no matter whether we're virtual or non-virtual. But I think thinking about team size and diversity, finding ways to engage faculty and students in a virtual environment, being clear on what the desired expectations and outcomes for these track 2 projects are. And I think starting to build strong jurisdictional support as well as cross-jurisdictional support for these efforts is important. I think as we think about years 3 and 4, we're really keen on increasing theme collaborations, continuing on active stakeholder engagement, as that's resulted to a lot of important networking opportunities for both students and faculty. And again, continuing to push this multimedia platform. So with that, I turn it back over to JD, and thanks again for the opportunity.
Great. So first thing that's most important, thank you to the speakers. It was an amazing set of talks. And I'd like to just close up just with a couple of just closing comments. The first thing is you can see that every single one of these projects is fueled by great science, hands down. But what you notice is there are common themes that underlie the science that are critical, right? You know, things like good communication, amazing resilience, right, of the scientific machine that allow projects to exist through COVID, through hurricanes and earthquakes. The value of students and their way that they're integrated cleverly to help do the work. Letting the young ones run the Ferrari. Sorry, Nick, I'm so going to steal that. Okay, but providing the young ones with the room to lead is critical. Standardization and development of materials across projects and across institutions becomes critical. The way that you make hiring practices and change hiring practices across your institutions can come in and be important. The idea of engaging early and often to build partners is critical. And so the thing that becomes important, and you've seen this already with the RII framework that Susie Iacono presented yesterday, right, is the idea of change within your jurisdictions or within projects and teams happens over a 3 to 10 year period. Surprise, surprise, the same length of time that one of these awards kicks in. Institutional change happens over 10 to 20 years. And jurisdictional change happens over 20 to 50 years. The point I want to make is that by going through these things, going through communication resilience, value of the students, letting the young ones lead, standardization and engagement, and most importantly, the one that's critical is having trust and inclusion in your projects. What that means is that these track twos that are on the scale of 4 years can have changes in your institutional culture that will result in the building and the building of competitive research within your jurisdictions. And that's an amazing thing to hold, an amazing thing to hold our projects to. So with that, please allow me to thank once again our speakers. I think it'll be pushing it if I allow questions, but please, by all means, feel free to reach out to any of these individuals. I know they're excited, and the way I view Track 2 is as a network of networks. You're part of that family, which is part of the bigger EPSCoR family as well. So Track 1s, if you want some advice, here's some great people to reach out to as well. Likewise, Track 4s, if you're looking for people to connect to, this is great. These are great opportunities. Thank you all so much. Thanks for hanging out for my session. I hope you enjoyed it. Take care and have an awesome rest of your day. Look forward to seeing you tomorrow.